When DNA is copied, it has two major ways to go. In the central dogma, we are going to talk about that. In one way, DNA produces RNA, which, if it is mRNA, directly produces protein, or if it is not mRNA, helps in producing protein. This protein, in turn, can have either structural or functional role in the cell. In another pathway, DNA reproduces itself, and this process is called replication. Transcription, on the other hand, is the process in which a single-stranded RNA is made from double-stranded DNA. When this RNA is messenger RNA, it is used as a pattern for making protein, and this process is called translation. Combination of translation and transcription is called gene expression. We do have another process here called reverse transcription. As the name implies, on the contrary of transcription, which makes RNA from DNA, here DNA copies are produced from RNA. As the dogma is crucial, I would like to work on that a bit more. DNA, through transcription, produces RNA, which in turn, through translation, produces protein. As we talked before, DNA also will reproduce itself through replication. What can be seen in blue is called gene expression, and what we see in green is replication. We also mentioned reverse transcription. As can be seen, it is making DNA from RNA. And as a general explanation, we should say that DNA has two ways to go: through replication and through gene expression. Through replication, it makes new version of itself which transfer to the daughter cells. And through gene expression, it makes all protein the cell requires. These proteins could be structural or functional. As we have the central dogma here, let's talk about some facts which will help us to have a better understanding of this dogma. First of all, genetic information is stored in base sequences in DNA. We talk a lot about gene. What is the definition of gene? We need the definition of gene at the level of step 1, not a very complicated one, the level that we need. Gene definition at the level that we need is a unit or sequence of the DNA that encode a particular protein or RNA molecule. And as we said before, gene expression is a mixture of transcription and translation. Here we have again some terms that we should be mastering them. As a matter of fact, a whole chapter is dedicated to some of them. First of all, DNA replication. DNA replication is the process in which each DNA molecule is duplicated before cell division. Needless to say, the whole DNA of the cell is duplicated almost at the same time. In a couple of minutes, we will take a look at cell cycle and we can have a better concept of this definition. Transcription. Transcription is transferring the information in double-stranded DNA molecule to single-stranded RNA molecule. Sometimes I think if the information in our brain is considered as DNA, our performance in an exam and the mark we get in that exam could be considered as RNA. And the process of exam, which the transferring or knowledge to the number is happening, could be called transcription. Maybe it helps for have a better understanding, maybe not. And the definition of translation. If RNA is mRNA, the information in base sequences of that converts to amino acid sequence, and this process is called translation. In the same way of the exam as I talked before, many times when your university sends out your transcription to other universities in another country, they should be translated. And finally, as you see in the chart, reverse transcription, the process in which DNA copies are produced from RNA. In retrovirus, it happens, and to a limited extent in some human cells. We will talk about that in some minutes, a bit more. Here we start talking about DNA, RNA, and their building blocks. The structure of nucleotides are an integral part of our discussion, since nucleotide is the building block of DNA as well as RNA. We have three main structures, which are our main concern here. First of all, nitrogen base, then sugar, and by sugar I mean pentose sugar. And the last part is phosphate group. Combination of nitrogen base and pentose sugar is called nucleoside, while the combination of all these three items, nitrogen base, sugar, and phosphate, is called nucleotide. We also can say that nucleoside plus phosphate group produces nucleotide. Let's have a closer look at each of them. First of all, nitrogen bases. They could simply be divided into two main groups. One group with one ring and the other one with two rings. The one with two rings are called purine, and the one with one ring is called pyrimidine. I think you have heard the name at least from high school. So we said nitrogen bases, based on the number of rings, are divided into two groups. Two rings are called purine, and one ring is called pyrimidine. So, we have two main purines in our discussion, which are adenine and guanine. We also have three main pyrimidine in our discussion, which are cytosine, uracil, and finally thymine. As you can see, the main structure of each group is more or less the same, and by main structure we usually mean the number of rings. The little difference in each group which makes each base identical is extremely important, and we will talk about them in much more details. Let's have a look at purine first. As we emphasized, for us at the level of step 1, the differences between bases are important. It is clear here that adenine contains an NH2 group at 12 o'clock, while guanine does not. This NH2 at 12 o'clock is the crucial hallmark here. Then we get to pyrimidines, the one with one rings. Again, cytosine with NH2 group. uracil without any group, and thymine with a methyl group. I should emphasize again that asking about the detail of structure in exam is highly unlikely to happen. However, the parts that make them distinguishable are crucial. Another point to mention here is that adenine, guanine, and cytosine could be found in both RNA and DNA, while uracil is just in RNA and thymine is just in DNA. The last point here is that we have another purine bases like xanthine, hypoxanthine, and uric acid, which are mostly concerned of other parts of biochem. So, let's have a look at the sugar part of nucleic acid structure. As we said before, this is pentose sugar. As the name implies, contain 5 carbons. We have 2 different pentose in nucleic acid. As you can see in the picture, the main difference between these two types is existing an OH or hydroxyl group in a certain carbon on the contrary of the other one, which has just hydrogen in that position. The one with OH is called ribose and is seen just in RNA, while the one without oxygen is called deoxyribose, and this one can be seen in DNA. Again, as the name implies, deoxyribose could be considered as the ribose without oxygen. I think here is a good opportunity to remember that RNA stands for ribonucleic acid, while DNA stands for deoxyribonucleic acid. So you can see that the difference in the pentose sugar leads to the difference in their name. It means the importance of this different in the name. In nitrogen base, the carbons are called based on their number. In order to avoid any confusion, carbon number in sugar contain prime. You can see an oxygen molecule on the top. Carbon 2 prime here is very important, as we mentioned before. Either hydroxyl group or a hydrogen will attach to this carbon 2 prime. If hydroxyl is here, the pentose is called ribose, which could be found in RNA. If hydrogen is here, the pentose is called deoxyribose, which in turn could be found in DNA. Here I would like to draw your attention to carbon 3 prime and carbon 5 prime. In some minutes, we will see the important role they play in the structure of DNA. Now it's time to talk about phosphate, which exists as a group, not as a molecule. If we have one phosphate in the group, this is called monophosphate. If it has 2 phosphate, it's called diphosphate. And finally, if there are 3 phosphate, they are called triphosphate. I would like to draw your attention to 3 important points regarding phosphate. First of all, the negative charges being carried by phosphate group. The second point is the high energy bound of phosphate. The last and very obvious one is the fact that nucleoside does not have phosphate. And when phosphate is attached, this is called nucleotide. Let's make a nucleoside. We want to make a schematic nucleoside. So we need a schematic nitrogen base. As you can see here, it has 2 rings. It could be 1 ring. We also need a schematic pentose, and the bond between these two is called glycosidic bond. But the type of this bond is not the concern of this lecture, and we will talk about that later on in another chapters. Now we have a schematic nucleoside, as simple as that. As you can see, a nitrogen base, pentose sugar. In order to have nucleotide, we have to attach a phosphate group to our nucleoside. But where? Let's have a look at these carbons. These are our 5 carbons. 3 of them are our concern in this discussion. As you can see, number 2', 3', and 5' are very important. Carbon 2' for determination if there is ribose or deoxyribose. We will talk about carbon 3' in some minutes. And carbon 5' is the one that attaches to phosphate group. Now we have a nucleotide, which could be called nucleoside monophosphate. As you see, if we say nucleotide, the number of phosphate group is not determined. While nucleoside monophosphate tells us just one phosphate group exists there. Please also remember that carbon 5' is the house of phosphate. Phosphate attaches there, and this is a very, very important point. Here again we have our nucleotide, which also could be called nucleoside monophosphate. So, when the next phosphate group comes, with this new phosphate group, it is still nucleotide, but no more monophosphate. This is now nucleoside diphosphate. In the same way, when we have the third phosphate group, it is still nucleotide, but nucleoside triphosphate. Here another issue comes up, the way these nucleotides or nucleoside are called. As a matter of fact, there is a simple rule for them all. Purine plus sugar will have the suffix of osine, and pyrimidine plus sugar will have the suffix of idine, osine, iodine. I know it's a bit confusing, but we try to practice in order to be more perfect with them. Based on what we can see here, you can assume that in any row you will see nitrogen base, then nucleoside of the same base, and 3 different nucleotides of the same base. The bottom line, the formula presents, which could be of help. Let's start with the purine like adenine. Depending on the pentose attached to that, in order to form nucleoside, it could be called adenosine or deoxyadenosine. In the same way, its nucleotide containing 1 phosphate would be adenosine monophosphate or deoxyadenosine monophosphate. If the number of phosphate group is 2, it would be adenosine diphosphate or deoxyadenosine diphosphate. And in this line, finally, our very close friend, very famous friend, very well-known friend, ATP, or adenosine triphosphate with ribose sugar, and deoxyadenosine triphosphate with deoxyribose sugar. In the second line, we have the other purine, guanine. The name of its nucleoside, as you can imagine, is guanosine when the sugar is ribose, and deoxyguanosine when the sugar is deoxyribose. In the same way as we talked, the nucleotide of guanine would be guanosine monophosphate and deoxyguanosine monophosphate, then guanosine diphosphate as well as deoxyguanosine diphosphate. When it comes to triphosphate group for either guanosine or deoxyguanosine, which would be guanosine triphosphate and deoxyguanosine triphosphate. After talking about purine, now turn comes to pyrimidines. Let's talk about cytosine first. With the help of the formula in the bottom line, we can assume that the name of the nucleoside with ribose would be cytidine and with deoxyribose would be deoxycytidine. In the same way, cytidine mono D and triphosphate and deoxycytidine mono D and triphosphate. Uracil, when binds to pentose, would be uridine and deoxyuridine. When we talk about glycogen metabolism pathway, you will hear about uridine a lot. Then, like before, for 1, 2, and 3 phosphate group attached to uridine, respectively, we say uridine mono, D, and triphosphate. Also for deoxyuridine, we call them deoxyuridine mono, D, and triphosphate. Remember that uridine is in RNA and not in DNA. The last pyrimidine nitrogen base we talk about here is thymine. Remember that thymine is found in DNA and not in RNA. In the USMLE book, as I searched, I just saw deoxythymine as the nucleoside. Deoxythymidine mono, D, and triphosphate are the name of nucleotide. So, deoxythymidine is the nucleoside, deoxythymidine monophosphate, deoxythymidine diphosphate, and deoxythymidine triphosphate. Here I want to use the opportunity telling you that DATP or deoxyribose adenosine triphosphate, DGDP or deoxyribose guanosine triphosphate, DCTP or deoxyribose cytidine triphosphate, and DTTP or deoxyribose thymidine triphosphate are used to build DNA, and we will talk about them in other chapters. Since we are here, let's talk about an integration with other discipline. We are going to talk about an enzyme called adenosine deaminase. Here we have adenosine. This amine group here is very important, and in a healthy person, this amine group is the target of this enzyme, adenosine deaminase. As the name implies, the amine group is removed by this enzyme. This new molecule is called inosine, and inosine here is not our concern. Deficiency in this enzyme can lead to a disease called ADA deficiency or adenosine deaminase deficiency. This is an autosomal recessive as well as a metabolic disease. As you know, most metabolic disorders are autosomal recessive, and it results in severe combined immunodeficiency. Sometimes it's called ADA-SCID. We have different problems which can lead to severe combined immunodeficiency, and ADA deficiency is one of them, and it accounts for 15% of all cases of severe combined immunodeficiency. In the picture, you can see skin of these people who have this disease. So, in the course of our lectures, let's talk about cell cycle in eukaryotic. This cycle is used in different disciplines. Here it is important for us since it reflects the timing of some crucial events in the cell. Let's start from the time that the cell is divided into two daughter cells. Since mitosis happens, it's called M phase because of the M at the beginning of mitosis. And the rest of the cycle is considered as interphase. Let's say interphase is the time between two mitoses. One of the most important characteristics of the whole interphase is the gene expression, including translation and transcription, which goes on throughout interphase. After M phase, or let's say after mitosis, the cell goes to gap 1, or simply G1 phase. In G1, the cell gets ready for DNA replication in S phase. S comes from DNA synthesis. After S phase, the cell enters gap 2 or G2 when the cell gets ready for mitosis or M phase. The mitosis is the one in which, as you see in the picture, the cell is divided into two daughter cells. As you know, some cells in the body do not divide. In other words, they do not have mitosis. So, no need to DNA replication and preparing for mitosis, meaning no need to S and G2 phases. In other words, there is no cell cycle for them. This special state of cell is called G0. Let's make some room in our diagram to show this state. The cells which do not divide are in this state. Some well known in this group are skeletal muscle, nerve cells, and the cell of liver or hepatocell. From another aspect, an example of cells which are existing in G1, S, G2, or let's say the cell which have cell cycle, a very good example of them is epithelial cell of intestine. The most important characteristics of G1 is the fact that cell gets ready for replication and DNA synthesis. Another important point to consider here is the fact that the cell cannot go from G1 to S phase on its own. This is a very, very important concept to know. From G1 to S phase, the cell cannot go on its own. Cell needs a kind of external stimulation or a signal, growth hormone, insulin-like growth factor, or PDGF or platelet-derived growth factor. They usually bind to a group of receptors called tyrosine kinase to induce their effect. In simple language, they remove barriers for replication. Then the cell enters the S phase for DNA replication. At the end of S phase, DNA content of each chromosome is doubled, and Each chromosome consists of two identical sister chromatids which are linked at the centromere. As you know, in interphase, we have gene expression. So, in S phase, we have gene expression and DNA replication. It means that in S phase, DNA goes to the both direction. Some RNA are made from it and also some DNA. Then we enter gap 2 phase or G2. In G2, replicated DNA is checked for any error. We also have cellular growth in G2, and other cell particles are divided before mitosis. I mean, DNA has been replicated, and we have two sets of DNA for mitosis, but some other particle of the cell should be also duplicated and divided before entering mitosis stage.